All right, well, welcome everybody. We'll probably have a few more people join us and they can just join us a couple minutes late. Just a couple housekeeping things, as I mentioned already, everyone's muted during the program. If you have any questions, please use the chat. You'll probably find it at the bottom of your screen. At the end of Dr. Jenkins' talk, we will address the questions and have a little bit of time for chat at the end. You can also use that just if you have any questions about Zoom in general. I'm happy to help. So this is the first of 4 talks on the science behind COVID-19. This has obviously been in the news, and there's every day it feels like we're all learning something new. So I'm hoping that through the process of these talks, we'll learn a little bit more about how our own bodies work, how COVID-19 works, and how that all interacts together. I'd like to give a special thank you to Ron Fanis for really putting this together. Thanks to his contacts and reaching out, we were able to get in touch with some really incredible scientists, including Marc Jenkins, who is the director of the Center for Immunology at the University of Minnesota. He is one of the foremost researchers in this field, and he was recently elected to the National Academy of Sciences. I think he said the first member of the School of Medicine there since 1970, I think. So an incredible achievement. We're so happy to have him here and very honored that he is joining us here with our friends and neighbors and people, I guess, near and far are here with us tonight. Yeah, so once again, if you have any questions, please feel free to use the chat. And with that, I'll hand it over to Dr. Jenkins.
Thank you, Teresa. I will share my screen here with you. Well, it's great. It's really great to be here. And it was great to hear from my old friend Ron Fanis, who was, when he was working for Boehringer Ingelheim, he was helped support our departmental retreat. And so I got to know Ron in those days. And I know he has some Minnesota background, and I know he was a hockey player. And so, of course, that resonates with Minnesotans. And so I was happy to accept his invitation to tell you about how your immune system responds to viral infection. And so my plan is to talk for about 30 minutes, leaving plenty of time for questions. I know this is clearly a hot topic these days, and we'll be talking about how the virus stimulates your immune system, and then how these vaccines work and how well they work, and then hopefully I can deal with some questions if you have any. So that's the plan. So really, basically, all you need to know about the immune system, at least the adaptive immune system, the immune system that can remember, the kind that we rely on for vaccination, can be summed up in what we call the clonal selection theory, which is a theory that describes the behavior of the set of lymphocytes that we have in our bodies. And this theory got the Nobel Prize for Macfarlane Burnet, an Australian, and it goes like this. And so you have stem cells, hematopoietic stem cells in your bone marrow that will go to either your bone marrow, stay in your bone marrow, or migrate to an organ. So it sits over your heart called the thymus. And those cells will differentiate into lymphocytes. And those lymphocytes produce, through a unique genetic mechanism, antigen receptors. And so in the case of B lymphocytes, that antigen receptor is an antibody. So we'll use that as our example. And so at least for the sake of argument, these stem cells proliferate, produce all these immature B lymphocytes, and each one expresses a different antigen receptor. And so imagine that the business end of this receptor, has a slightly different shape than this receptor, which has a slightly different shape than this receptor, etc. Repeated about 100 billion times. So you can produce a lot of B lymphocytes in your body. Now, the process that generates these antigen receptors is, at least for the sake, as best we understand it, is random. And so these ends of these antibodies are slightly different, and they're slightly different through a process that generates a lot of random shapes. And so there's a good chance that some of these antigen receptors will be able to bind to some of our own molecules. And this would be bad, because if we allow those lymphocytes to become fully activated, proliferate, do their job, we would have autoimmune disease. So when lymphocytes are immature, if their antigen receptor binds tightly to any substance, that cell is killed. And so many, quote, self-reactive cells are lost from the repertoire. So, and we think it could be as many as 90% of the developing lymphocytes are eliminated because they are self-reactive. They by chance created a self-reactive receptor. So maybe we go from 100 billion cells down to 10 billion. That's still a lot of different randomly shaped receptors. And so the cells that survive this process of deletion of self-reactive cells leave the bone marrow, and they go to our lymph nodes. And so you have about 1,000 lymph nodes in your body. They're kind of just under your skin or near some of your internal organs, and they're collecting lymph from the surrounding tissue. And we'll come back to that idea in a minute, because these lymph nodes are really important sites of immunity, where immune responses begin. So your 10 billion or so lymphocytes are moving around between your lymph nodes all the time. And if you get an infection, the hope is that by chance, you will have some cells that have receptors that can bind to that foreign substance. So if we think of the coronavirus, you will by chance have a few cells in your body that can bind to proteins or molecules on the coronavirus. Those cells then get a signal through their receptor, which, unlike in the immature state, that signal causes the cell to die. When the cell is mature, if it gets a signal through its antigen receptor, it divides and reproduces itself 1,000 to 10,000 times. And that happens in lymph nodes, but these cells that have proliferated in response to this antigen can migrate from the lymph nodes into sites of infection and help to clear the infection. And so a really important point is that we all had lymphocytes in our bodies that had antigen receptors that are specific for elements of the coronavirus before the coronavirus epidemic. And that's an important concept. So your immune system is basically like a big casino. Make 10 billion different locks and hope by chance that when a new key comes into the body, it will fit into a couple of those locks, and then reproduce all those locks to produce lots of cells that are specific for that key and eliminate it from the body. So that's the clonal selection theory in a nutshell. And it applies to 3 different kinds of lymphocytes and you're maybe reading about some of these lymphocytes in the paper. So I told you about B lymphocytes. Their antigen receptors are antibodies, and they can basically bind to anything. Any foreign molecule that comes into your body, you can usually make an antibody against it. And then there are these two kinds of T lymphocytes called helper T cells. So they undergo this clonal selection process, but they are restricted to recognition of fragments of proteins on the surface of our own cells. And I think you can imagine how that becomes important, especially when we think about these killer T cells. Because if a cell is infected with a virus and has become a virus factory, some of the protein fragments from that virus will come to the surface. Killer T cells, a few of them from the repertoire, will be able to recognize that protein fragment and kill that cell. Now, that may sound like a bad idea. In the case of coronavirus, that's one of the epithelial cells in your lungs, but it's to your advantage to kill that cell and prevent it from producing more virus. And then there are these other T cells called helper T cells. They also bind to their receptors, detect cell-bound protein fragments, and they provide signals that B lymphocytes and killer T cells need to do their job appropriately. So that's, those are the three kind of lymphocytes. They all obey the clonal selection theory in that they are generated, they're randomly, the receptors are generated randomly, the self-reactive ones are eliminated, and the foreign reactive ones then are the source from which a few will be specific for any foreign thing that comes in your body and will be the movers and shakers for your immune response and your ability to clear that infection and then have lifelong immunity to that infection. And so that raises another aspect of the clonal selection theory. So if we imagine, this was the coronavirus and it stimulated the coronavirus specific cells, lymphocytes, after an initial infection, and you survive that infection, instead of having, one cell like this, now you have a huge army of cells like this. And some of these can survive for the rest of your life. So that right there tells you how immunological memory works. You expand and retain the cells, the lymphocytes from your past infections that can protect you from future infections. So let's expand out on a little bit on that and talk in a little detail about coronaviruses. Specifically now, the coronavirus that is now causing the pandemic is called Sars Cov2. And this is an RNA virus, which means it has an RNA for its genome. And then that RNA is coated with some other proteins. And a famous one is called the spike. And the virus uses that spike protein to bind to a receptor on epithelial cells in our lungs. And that receptor is called the ACE2 receptor. So when that happens, the virus is internalized by that lung cell. And then it breaks out of this vesicle, uncoats its genome, which is RNA. That gets translated into these proteins, spike and the M, that then coat that genome, and a new virus buds out. And of course, a single infected cell in your lungs can produce thousands of viral particles. So your immune response detects this through sentinel cells called antigen presenting cells, sometimes also called dendritic cells, that take up the virus and then chop it up into these protein fragments. And so here's a protein fragment from the virus, and that can alarm a helper T cell that has a complementary antigen receptor. And then that helper T cell can help B cells that have coronavirus specific antibodies as their antigen receptor, and killer T cells that can also recognize protein fragments from the virus. And so these B cells, when they get that signal from that helper T cell, they proliferate and they start to secrete their antigen receptor. They start secreting their antibody into the body fluids. And these antibodies can then gum up the surface of the coronavirus, binding to the spike and preventing the spike from interacting with ACE2 and preventing infection. Now, some of these B cells will survive for the rest of your life, at least in theory. And some of them retain the antibody on their surface and some of them secrete the antibody. So you'll have coronavirus specific antibodies in your blood for very long periods of time. And you will have what we call memory B cells in your blood for a long time. So if you get a second infection, those memory B cells will now start secreting their antigen receptor, making more antibody, and you boost the amount of antibody in your blood. And then, as I mentioned earlier, these killer T cells that have antigen receptors that recognize the protein fragments from the virus can actually start killing the infected epithelial cells to try to stop virus production that way. So the combination of these immune mechanisms will, if you're lucky, eliminate the virus from your body and then produce these long-lived memory cells in expanded numbers that can respond very quickly to a second infection, so quickly that you don't even know you were infected. So let's look at that another way, and let's look at that over time. So if you get a SARS-CoV-2 infection, these sentinel cells, antigen-presenting cells, or dendritic cells, take up that virus. They make those viral protein fragments, and they stimulate the killer T cells, the helper T cells, shown in two different flavors here, which help the B cells to make antibodies and to form memory T and B cells. And again, these cells all have antigen receptors that are specific for proteins and molecules from SARS-CoV-2. So these antibodies and these cells are useless for flu infections, strep infections, staph infections. They are only good for SARS-CoV-2, which induced them. And so if we look at this over time and we track here infection, which plays out over weeks, it goes up and it comes down, and it comes down because of your adaptive immune system. You will start to make specific T cells will start proliferating, and you will start to make antibodies within about 10 days after the infection. And then those T cells will, and B cells and antibodies will clear the infection and then return to some lower level that is lower than the peak, but higher than the starting point. And these antibodies and memory cells can last for years. And in fact, most of us who got the polio vaccine when we were kids still have polio specific antibodies in our blood, in my case now, 60 years later. So that's what we call immunological memory. And so that's the process that we're hoping to induce with a vaccine. So how long does the immunity induced by the infection last? And so that's been in the news a lot. And so here's some actual data from this paper that was published in Science. And so on this axis, we're looking at the amount of spike protein specific antibody. And you can see in people before the infection, they don't have very much of this. And this is a log scale. So in these units, this is 10, 100, 1,000. And so after the people get infected and they have symptoms come on, they make antibodies. And that line is essentially flat for 240 days. So 8 months. No evidence of decay of the antibody. So that's good news. That fits with what we know about a lot of other viral infections: mumps, measles, rubella, polio virus. They can induce lifelong immunity via antibodies. Because remember, these antibodies are against the spike. And so if this person gets infected here at day, out at day 210, so 7 months later, they will have, this much antibody to bind up that spike, prevent it from interacting with ACE2, preventing the infection. And an immunologically naive person has, 100 to, more like probably 10,000 times less antibody. And so is susceptible to disease because it takes time for the host to make enough antibody to clear the infection. And that's why usually when we get a viral infection, even though it induces a good immune response, we get sick because that first immune response takes time and it doesn't come up quick enough. Whereas that second immune response starts from a much higher number of specific T, B cells and antibodies. And so you can eliminate the infection before you have any symptoms. So I don't know how many of you, most of you I'm sure had the chicken pox when you were a kid. And I was the oldest kid in my family. And so I had the chicken pox first, and then all my brothers got the chicken pox, 2, 4, 6 years later. And of course I didn't get sick, but I guarantee you I got infected with them slobbering all over me all the time. So, but I had this kind of immunity set up, so I didn't get sick. Okay, so what are these vaccines? So remember, the coronavirus is an RNA vaccine, meaning its genetic material is RNA. And when it enters cells, that RNA gets translated to make its proteins so that they can assemble with that replicated RNA genome to produce a lot more virus. So what the vaccines are, at least the Pfizer and Moderna vaccines, are to take the messenger RNA that encodes the spike protein and then wrap that in a lipid shell to make it kind of like a virus particle, and then inject that into our arm. And that RNA, via that lipid shell, will be delivered inside these dendritic cells, those antigen presenting cells, sentinel cells, and they will start to make the spike protein, but only the spike protein, not the rest of the proteins. So the virus cannot actually form and you can't get the infection from that vaccine. But the spike is foreign protein from the tenets of the clonal selection theory. We know that you will have T and B cells that will recognize it. And that recognition will initially occur in lymph nodes that are in your armpit, because the lymph that goes from your arm runs to a lymph node in your armpit. And so that's where the T and the B cells will become activated. The B cells will start making antibody. The killer T cells will migrate to your lungs and start killing infected cells. And antibodies go into your blood. So even if they're made in a lymph node in your armpit, they go into your blood and then they're everywhere. And they can access the lung tissue and start gumming up the virus at that point. And so, you could get the vaccine in your thigh muscle, in your gluteus maximus. Different lymph nodes would drive the response, but the effect would be the same. The shot goes into the arm because it's convenient. I don't know about you, but I'm more willing to bear my arm in public than I am my butt. And so it's a convenient and efficient way to induce immune response, because the needle goes into the muscle. There's lymphatic vessels, will take the vaccine to the lymph nodes, that draining lymph node, and you will get the immune response cranking. And of course, these vaccines have this prime boost modality. So, if you get the Moderna vaccine, you have to go back a month later. So the idea there is that you've gone about a month, and so you've got this initial spike of antibody. T and B cells have now come back down, and you've got some of these memory B cells. So when you get the vaccine again, they will differentiate into antibody-forming cells, and now you'll get another big boost of antibody that will produce a set point that's way up here. So that's the idea behind the boost. It's like reactivating those memory cells, but only a month later. Whereas, like when I got the chicken pox, my memory B cells got reactivated probably 2 years later when my next brother got chicken pox. So we're compressing that time frame. So here I thought I'd show you some data from the actual Moderna clinical trial data that was published in the New England Journal of Medicine. And so here we're looking, basically this is the amount of antibody that people made with an arbitrary unit. Again, this interval is 10 times more than this one, which is 10 times more than this one, which is 10 times more than this one. And so these were 3 different vaccine doses. And so after the first shot, you look 1 day later and people basically have this background amount of antibody, let's call it 10 units. And then by about 2 weeks, you can see this major rise to about 2,000 units, which stays about at that level until the time of the boost, which happens here. And you go from 2,000 to 3,000 units up to about 50,000 units that is then maintained for a couple of weeks at least. And now, we know from other studies that, the antibody levels are staying pretty constant, at least for 5 or 6 months, probably a lot longer than that. And interestingly, if you compare, and I believe this is the actual dose that's in the vaccine, you can see that people end up after their second shot with more antibody on average than people who recover from the infection. So these vaccines are inducing an incredible amount of antibody, which is a good thing for our immunity, because neutralizing that spike protein and preventing it from getting to the ACE2 receptor is a big, big reason that we get immunity. So how good are the vaccines? And so, we hear about, they're 95% effective. I think the Israelis have done a nice study that I think illustrates this point pretty well. So, they were way out in front of this, so they got a lot of people vaccinated quickly. And so in the observation period of their study, they looked at, 800,000 people who weren't vaccinated and about 400,000 who were. And they got 12,944 infections in this group and 254 in this group. And these were all very mild infections. So per 10,000 people, the infection rate was 166 per 10,000 in the unvaccinated group. And in the vaccinated group, it was 6. So it reduced infection risk by about 30-fold. I think there's gathering evidence that the vaccine is reducing the risk of death by more than that. But it's reducing the risk of infection by at least 30-fold. And you can see that in the course of their trial as they were studying people, after the second dose, they actually got a few people who had contracted the virus, but by 3 weeks after that, they really didn't find any infections. And so, the vaccines are working really, really well, but they're not perfect. There are some breakthrough infections. And so, I like to think about it in terms of relative risk. So if we think about the last year, we've had about 30 million infections in the United States from Sars Cov2 in a country of roughly 300 million people. So the annual risk of infection was 1 in 10. That's remarkable, And of course, that's average across the whole population. So, if you were older, this number would be higher, risk was greater. If you're younger, it'd be less, but that was the average. And the death, so about 600,000 people died from the coronavirus infection. That's 1 in 500. That's a remarkable death rate. And to compare that, your risk of dying in a car accident in 1 year is 1 in 48,000. So this risk was 100 times greater than dying in a car accident. So that's a serious risk. So if we said the vaccine, if everyone had been vaccinated in that period, that risk would have been reduced by 30-fold to 1 in 15,000, which is that would be about 20,000 deaths from coronavirus in that period, which is about the number of homicides that occur in the country. So the vaccine is taking your risk from some risk that's greater than any other kind of day-to-day risk you would ever face in a normal year to something that's in the range of something that most of us don't think about every day. And although some of us, it depends on what group you're in these days, but consider to be an acceptable risk. So, and if you go to actuarial tables, you can find other risks that are in this range. We have about 20,000 deaths from influenza virus every year. So in the vaccinated population, we would have reduced the risk from Sars Cov2 to something in the range of seasonal influenza. And of course, society accepts that risk, without masks. And, most of us lived our lives fully without masks, and accepted that risk of having a fatal flu infection. So maybe I find now that kind of risk assessment helpful, maybe you don't, but but I thought I would throw that out there. So that's all I got. I would like to, I think I'll leave this slide up. Actually, I think I'm just going to stop sharing my screen and just talk to people. So there's questions.
Yeah, so there's, thank you for that. That was really interesting. I had actually always wondered why, and you kind of talked about it, why we now get shots in the arm when it used to be so much, not in the arm.
Yes, for the reasons I said.
Yeah.
It's a matter of practical. It's a practical matter. There's no difference in a single intramuscular injection in your arm versus your gluteus maximus.
I guess it's more fun to portray in books and movies and TV the other way, but
yeah,
we did get some
dramatic,
reasonable questions other than that one. In the chat, someone asked, when in evolution did the clonal selection process of immunology develop? Are there any other biological processes that are similar to clonal selection?
Yeah, well, that's a fantastic question. So we know jawless fish, so like lampreys, are the first animals in ontogeny that have lymphocytes that would obey the clonal selection theory. So this is an ancient form of immunity. Now, if you go further back than that to like insects do have a form of immunity, but it doesn't involve lymphocytes. It involves phagocytes. We call that innate immunity. We have innate immunity too, but innate immunity is good if you want to live for a week. If you want to live for longer than that, you need adaptive immunity. So that's a very good question. I think a great analogy for the clonal selection theory is actually Darwin's theory, where of evolution on a much larger scale. So you think about a huge population producing mutations randomly in the population, and some of those mutations improve fitness, and those mutations get selected and become enriched in the population. So in the same way, lymphocyte receptors are generated randomly, and the ones that are fit are selected by the antigen and are enriched in the population. So the process, though, where antigen receptors are randomly created in single cells is unique to lymphocytes.
Nice, thank you. And the second part of that same person's question is there any known role for exosomes in the viral infection process or in the immune response to the virus? I can't speak.
Yeah. Okay. So this is a very in the know questioner. So exosomes are fragments of our own cells that can bud off and be taken up by other cells. So you could imagine an infected lung epithelial cell could have a little bit of its membrane bud off with some virus in it, and then that could get taken up by dendritic cells and sentinel cells and stimulate this process of immunity. I'm not aware of that going on. That's been observed in other viral infections. It probably is going on. Its significance in the grand scheme is not clear to me.
Thank you. Another question, does a mother who received the polio vaccine, for example, pass those antibodies to her biological children?
Yes. So antibodies can cross the placenta and enter the fetus. And for those of you who have had little kids at home, you may have noticed that your baby's got kind of about a 6-week grace period, or they really don't get many infections because they have acquired all the antibody memory from the mother. So all the mother's past infections and past vaccinations. But because the fetus doesn't have its own lymphocytes making those antibodies, it just acquired the antibody proteins themselves. They will eventually disappear. And so when that baby gets to about 6 to 8 weeks, suddenly their nose is running the whole time because they're now going to they are basically an immunological blank slate and they have to run through all those infections themselves. And so our hope is we vaccinate those kids so they don't have to get the infection itself and risk their life.
Yeah, that's interesting. How do Moderna and Pfizer produce the spike mRNA at scale?
Well, that is one of the main reasons that RNA vaccines were really thought to be the way to go at first, even though they're relatively untested. RNA is relatively cheap to make, so you can make bacteria produce that messenger RNA, and you can then purify that messenger RNA. And so in massive fermenters, you can make lots of RNA. And because when the RNA goes into cells, each RNA can produce many protein copies, you get an amplification effect. So, but if you recall, it took them a while to ramp it up, because they had to ramp up at a level, that we've never seen before. We try to get everyone in the population, vaccinated against, get their DPT shot, but that's a small number of kids who are born every year, they get their shot, right? We're not trying to vaccinate every person in the country at the same time. So it was a huge undertaking.
Is the magnitude of T-cell responses measured for COVID, and how important is it versus antibody response?
Yeah, we don't know. We don't know. In fact, we're assuming that the mechanism of protection is antibody. And that's because some of these antibodies will prevent viral particles from binding to ACE2 and entering cells, so-called neutralizing antibodies. But frankly, that's a hypothesis because T cells are generated in massive numbers too. And we measure them by taking some blood cells and then adding viral fragments, those viral protein fragments, and then measuring what are called cytokines, which are immune hormones made by those T cells. And then the amount of that cytokine is proportional to the number of T cells that are specific for that protein fragment. So those assays are pretty good, and we know that this virus stimulates a massive helper and killer T cell response.
Good. Someone asked, my husband and I had the virus mild. We both had the Pfizer vaccine and I had more reaction than he did. Is there a reason why?
Yes, and the reaction is almost certainly related to your immune response. So, probably having a reaction is evidence you have had an immune response to the vaccine, but we're not really sure. My guess is that reaction comes from T lymphocytes that are sitting there actually in the muscle at the injection site. They were generated by, in your case, your original infection. They proliferate in your lymph nodes, but then they go out and start surveying all over your body. So they're sitting there. When you get that injection, which would be the second exposure for you, those T cells make those cytokines at the injection site, and those cytokines are what cause the symptoms. Now, that doesn't necessarily mean that someone who doesn't get sore as sick at their injection site won't also have immunity, because that's probably related to the B cells. And we know that, at least anecdotally, that many people who don't have serious reactions have high levels of antibody and protective immunity. So it's probably something to do with you probably had more T cells at your injection site than he did. That's what I would guess.
I probably had a lot because I felt awful after my.
Yeah. Well, those, your immune system is really designed to kill things. And so it does it usually in small amounts and locally. So if you get a systemic immune response, it can make you pretty sick.
Yeah. Well, I had a nice cuddle on the couch with my dog for a day. That was
luckily it's transient.
Someone asked, do viruses ever mutate to less virulent but more dominant forms? Also, can you comment on how the 1918 flu died out?
Yeah.
Virulent?
Yeah. Yes. Virulent. Yes. So, most mutations are deleterious to the virus. It's already got a genome that works. So when you start messing with it, most of the changes are bad. So those viruses are less fit. And so, and viruses are trying not to mutate to forms that kill the host too fast. Because if the host is dead too fast, that host is not walking around spreading infection. So the virus is trying to find some sweet spot where it's infective enough to infect a lot of people, but not kill them too fast, or the infection can't spread far enough. That's why when you look at Ebola burns out so fast because it kills people quickly. And usually the infection will burn out because the susceptible hosts are dead and the infection stops. So it's trying to find this sweet spot. Why the flu in 1918 flu died out, it's not completely clear. It may have basically killed or infected all the susceptible people in the world, and then everybody who was left had immunity. So again, a dramatic example of natural selection.
Yeah. Someone asked, is natural immunity better than vaccine-induced immunity?
Probably not, because the amount of virus that different people who get an infection varies greatly. And so my feeling is, if you came within an inch of death and survived, you've got phenomenal immunity. But most people get a much milder infection. And that's probably why in that graph I showed you, the amount of antibody on average in people with natural infection was lower than people who get the vaccine, because people get the vaccine, get the exact same dose and the exact same regimen, 3 or 4 weeks apart. So I think on average, the vaccine is probably better, because if you have natural infection, you might have had a mild infection that gave you less antibodies.
And And can you talk a little bit about the booster shot that Pfizer and Moderna are talking about? And we haven't heard anything about Johnson & Johnson, but
yeah. Well, Johnson & Johnson is a DNA vaccine. It's effective adenovirus. Again, it's working this kind of the same way. It goes into cells, and then the DNA gets transcribed into RNA that gets translated into protein. So there's one additional step, but it's a single shot vaccine, basically because they decided to do their trial that way. It's not clear that the boosts are really needed for immunity. That's the way the trial was done, and we know that works really well. So we now have 8 million people I heard recently who have chosen not to get their boost. They're taking some risk there because that's not how the trial was done. But if you remember that graph I showed you, one shot gives you a lot of antibody. The question is that boost that's going to give you 20 times more antibody than that worth it? And I, as a betting, if I'm betting, I'll take that extra 20-fold. So that now the purpose of a third boost might be to deal with variants. And there are certain viral mutations that have gained in fitness. They're more infective, at least mildly so, and they may have changed their spike enough to evade immunity. This is clearly going on, but I think we already know that I don't think that the UK or South African variants are going to just decimate the country, because if they had that capacity, we would have had massive outbreaks in nursing homes again amongst vaccinated people, and we're just not seeing that. So I think the idea is, though, there could be a variant that could arise that's even worse, has more immune evade, is a more changed spike from the original one in the vaccine, and now that immunity won't work anymore. So the idea is, let's get out in front of that and be ready with a boost for that variant on the horizon. So I think that's probably the thinking that's going on there.
Okay. Someone had a question. I might kind of put something in. Someone asked about monoclonal antibody treatment. Our speaker, I'm going to plug our speaker next week at this point, is Dr. Benjamin Fulton with Regeneron, who's going to talk about antibody therapy.
That's perfect. That's great. Ron got some serious talent for you on that, because Regeneron is the leading monoclonal antibody company. And so monoclonal antibodies is a technology where you fish out a single B cell that is secreting, in this case, a spike-specific antibody, and you immortalize it. You turn it into a cancer cell, so you can grow it forever. And it will secrete antibody at huge amounts. And you can then grow vats and vats of that one antibody, so that you could infuse that into someone who doesn't have their own immunity. And it can provide protection. So that's what monoclonal antibody therapy is.
Okay. I'm gonna start kind of picking questions here so we don't go too long. Can you explain the new T-Detect test?
No.
Okay. That's a quick answer.
I don't know what it is, so okay. I can't.
Let's see. Is there evidence that those with asthma are not as ultra susceptible to COVID as might be predicted? Yeah.
There was a recent paper that looked at that, and the surprise was that the chance of serious infection in asthmatics was really not higher than the general population, which was surprising given that they have some airway issues. So that was a surprise.
Let's see. Someone asked, they had a severe infection of COVID-19. Should they still get vaccinated?
Yeah, that's a tough question. I think what we know, what do we know? We know that people from the clinical trial data of incredible high quality, that people who get 2 shots of the vaccine have protection. We don't know that for anyone else. We'd be speculating. And there were, I guarantee you, there were some people who had prior COVID infection who were part of the trial. So anything else is speculation. And I really wouldn't want to go there. So I think you should get, I would go with what we know.
Can you comment on so-called long haulers and what might be going on there?
Yeah, I don't think we have a clue what's going on there. And you know, that syndrome, you know, chronic fatigue syndrome, other things like this have been described after other kinds of infections. They've been poo-pooed by clinical medicine as psychosomatic illness and other things like that. That needs to be really readdressed. I think that it's very certainly, there's a biological basis to that, and a ton of research needs to be done to see what's going on there. The virus causes a lot of damage to blood vessels. And of course, we have blood vessels in all of our tissues, all of our organs. And so this kind of multi-organ manifestation of that long haul syndrome tells me that it's probably something related to blood vessel, what we call microvascular injury. But, you know, there's really no really good evidence one way or the other on that problem. But clearly, with, you know, 32 million infections in the country, this is going to be a big deal.
Yeah, learning something new every day. I'm going to plug another one of our talks in this series. Someone had a question about stem cell research in the development of vaccines. On May 12th, our 3rd talk, we're having Dr. Cyril Kuhn from Boehringer Ingelheim. I can't speak anymore today. I'm sorry, everybody. But anyway, he's going to be talking about drug discovery. So that might be a good time to address that question. Hopefully that person can join us for that one too. Let me see if there's any other questions on here. I think I might have gotten everyone.
I would like to answer one there that I see, Teresa, from Michael.
Okay. Oh,
many people seem reluctant to get vaccinated because they are experimental. Why is this, and what is needed to fully approve? And that's a really good question. These vaccines have undergone the same kind of safety trials that all vaccines have undergone. And that was especially important for the RNA vaccines because these are first in class. You know, these kinds of vaccines have never existed. And so really the only difference, and during the trial, all what are called adverse effects have to be reported by the people doing the trial to the FDA, and they get investigated. And we've heard some trials are halted because adverse effects were reported and FDA said stop. And so these trials got emergency use authorization, which is not full FDA approval, just given the pressing nature of the pandemic. But so the main difference between these trials and what would be done for other kinds of vaccines is the number of people who are in the trial. Because if you, like, there were about 50,000 people-ish in the Moderna and Pfizer trials. If you have a side effect that occurs at a level of, let's say, 1 in 500,000, you can't detect it in a trial with 50,000 people. You need 500,000 people to find one of those kind of adverse effects. And so, you know, so if there's any, so that's the level of concern. Could there be a side effect that exists at some level greater than 1 in 50,000 that could rear its ugly head? Well, there could have been, I guess, but the bottom line, we just did the experiment, vaccines in 200 million people. And, if there were massive, people are growing two heads, you'd have read about it in the New York Times. So there was some risk there, very small, but we got by that risk. And it's just remarkable to me how well these vaccines work, given that they were untested technology.
Okay. One last question from someone who knows a lot more words than I do. How is the adenovirus disabled or attenuated for its use in vaccines? How risky is use of such viruses as vehicles for the viral DNA?
Yep, good question. Well, these kind of vaccines, adenoviral vector vaccines, these have been around for a while. We have used these, so we know more about their safety profile. It's basically the DNA. It's a segment of DNA that gets packaged in a defective adenoviral particle, so the virus can't replicate, and that small piece of DNA then gets translated into a messenger RNA that gets translated into the spike protein. So the safety profile on these kinds of vaccines in other vaccines of this kind has been very good.
All right, well, I think that's it. So I want to say a huge thank you for joining us this evening. I'm letting everybody unmute themselves now if they want to say hi, say thank you. Thank you so much for joining us all the way from Minnesota here in Connecticut. So we really appreciate it. I personally learned a lot. It's been a long time since I took a science course. So this has been really interesting. So thank you so much.
Thank you, Dr.
Thanks.
Jenkins.
for obtaining
Mark,
some wonderful
For
people.
me personally,
Thank you.
thank you very much. And we'll get together for a beer and a brat when I'm there in September.
All right,
Oh.
Ron, I'll take you up on that, man.
Okay. Maybe 2 beers.
Yeah, oh yeah, okay. Twist my arm. Bye, everybody.
Bye.
Thank you so
Bye.
much.
Thank you.
Bye.